Hello and welcome, dear friends. We move on to the last point of discussion in this unit, and the point is bacterial transcriptional and translational cycle. There exists a marked difference in protein synthesis of eukaryotes and protein synthesis of bacteria, prokaryotes. Though we have been seeing majorly the prokaryotic protein synthesis, at this point of time, I would like to raise the bar of your understanding a bit higher. That is what graduation is all about. Gradually, step by step, you should move towards a level of higher understanding. And the more you dwell into the molecular level of the processes, more deeper insights you will acquire with respect to the phenomenons governing life. And when you start to understand the phenomenons governing life, then you have started to, or you have just begun to understand what science is all about. So in this last lecture of this first unit, I would like to raise the bar a bit and take you into a domain, into an area of bacterial transcriptional and translational cycle, where some magic proteins, they govern a very efficient process in prokaryotes. Why we need to read and study prokaryotic systems? That always has been a question. Why to study such a primitive organism where such highly evolved systems in humans and eukaryotes are there? That is because, most importantly, each and every cell needs to function with the help of proteins. If you see this slide, if you just see the slide over here, see this network, each and every single dot here is a protein. So let us say this is a proteomic map of a cell, any cell in general. This is a picture, you will find it in Introduction to Genetic Analysis, 10th edition by Benjamin. That is the most important what you can say part in any life is protein synthesis, and complete set of proteins produced by genetic material of an organism. Let us say if it is shown like this, and the dotting lines are how this one protein interacts with another protein, or how this one protein is necessarily signaling the functioning of another protein, is shown by the lines, and we will call that as an interactome. Many instruments like HPLC, gas chromatography, bioinformatics, they are used to create such maps. This is just to give you a broad idea of how one single cell functions. So a complete set of proteins produced by genetic material of the organism, a proteome and its interactome, complete set of proteins interacting in an organism. If it is like this, now think about these cells with interacting with one another. Hundreds, thousands and millions of such cells forming a tissue, such tissues forming an organ, and an organ forming organism, and everything at the level of DNA. So it becomes more and more complicated, interesting, fascinating. So when you hear the word complicated or a complex, the next word which you should come to your mind should be interesting. So that is where our whole system goes wrong. When we say the things are complex, suddenly you think I will not get it. No, when you say the things are complex, then things are more interesting, more fascinating. So one such fascinating area is the bacterial transcription and translation, which is going on simultaneously. See here, I have given you this figure to cement in your brains. Make this is my own pictures which I have drawn in paintbrush, and I want you to fix this in your brain. This is transcription, the central dogma of any protein synthesis. This is transcription. DNA, RNA polymerase and mRNA being formed. Second step, translation. mRNA, ribosomes, tRNAs accumulating the amino acids forming a polypeptide chain. Now in eukaryotes, these two are separate processes. Whereas if you see in prokaryote, come over here. Now fix this picture in your mind. For bacteria, microbiologists, transcription going on simultaneously, a translation has started. Okay, 5' to 3' and 3' to 5' and polarities for transcription, translation All these are maintained. How this process functions and how this is very important, not only for survival of a bacterial cell, but for us to understand the mechanisms of evolution. This is a beautifully, what you can say, synchronized process. Homeostasis is there. What is homeostasis? I will explain that. Homeostasis of nucleotides and the amino acids. A synchrony. No errors are there. Perfect, what you can say, harmony. In perfect harmony, this transcription and translation takes place in bacteria. Of course, elongation, initiation and termination is there. So what happens at the molecular level? So as I said, I am going to raise the bar a bit. So this may find a bit tough for the graduate students, but postgraduate students will understand. But as graduates ultimately will aim for becoming postgraduates, I think it is important for you also to understand. Otherwise, take as much as you understand, read, try to explore, have a discussion with me, and then you will step up to the next level of genetics at the molecular level. So come, let's see the CTT, coupled transcription and translation in bacteria. On the left there is a prokaryotic cell, on the right there is a eukaryotic cell. Now see here, this simple, in a very simple perspective, just observe, blue line is DNA, pink line is mRNA. Okay, from the DNA, mRNA transcription is taking place, and simultaneously a polysomal structure, that is one mRNA with multiple ribosomes, is shown. That is what is going out in a prokaryote. So this is called as CTT, or coupled transcription and translation. Shift your focus now to eukaryotes. There is a nuclear membrane. E. coli is the model organism. Whatever the studies, pictures, photographs, they are mostly with respect to E. coli, but it is the best known representative for a prokaryote. And in this prokaryotic system, you don't have a nuclear membrane, you don't have boundaries. The cell has chosen not to keep any boundaries. So what? So the whole process becomes very fast, rapid and efficient. Simultaneously transcription is going on, translation doesn't wait for the transcription to get completed, and you can see translation has started. Things change when it is a eukaryote. It is complex, it is large, huge. Evolution has taken its own course of time in managing the molecules at the genetic level. What happens here is, there is a nuclear membrane. These are nuclear pores. The gaps are nuclear pores. DNA, the blue line, synthesizes a mRNA, which is not a final transcript. The dark shades here, you can see, those are the introns. These are the non-coding sequences. Focus on what I am saying. These non-coding sequences are removed. Exons are joined. Introns are removed. This is called as a post-transcriptional processing. Doesn't take place much in the prokaryotes, but it takes place in the eukaryotes. So a post-transcriptional processing brings out a shorter mRNA where introns, non-coding regions, are removed. So you have a shorter mRNA transcript. That transcript now moves outside the nuclear membrane through the nuclear pores. One of the animation in the links, you can see that, of WEHI education, WEHI education, beautifully shown animation. The mRNA moves through the pore, comes out in the cytoplasm. In the cytoplasm, translation is taking place. So it is not coupled. It is not a coupled process. What is happening there is, the whole system is compartmentalized, whole process is compartmentalized. Transcription is taking place inside the nuclear membrane, translation is taking place outside the nuclear membrane. There is a post-transcriptional processing. It involves 3 to 4 steps. I will just mention. There is a 5-methylguanosine cap, 5-MEG, for the mRNA, and a polyadenylate tail is there in the mRNA end. So 5-methylguanosine cap and a poly-A tail is added. Then it moves outside to the cytoplasm through the nuclear pores, and their translation takes place with the larger and the smaller subunits. Now we have seen the differences of a eukaryotic ribosome, prokaryotic ribosome. Rest processes are same. So a more detailed picture you can see here. This is what is happening in prokaryotes. A DNA translation is highlighted, transcription is simultaneously going on, and charged tRNAs, they are continuously bringing the amino acids even as the transcription has not ended. Now this gives a very good chance for the prokaryotes to control or regulate the protein synthesis. I am giving you a thread here. It has to be linked into next lecture when I will be talking about regulation of gene expression. This coupled transcription and translation plays a very important role in the regulation of gene expression, specifically tryptophan operon. Operon, what is operon? Any gene when it is transcribing its segment into proteins, and how much proteins need to be synthesized, the cell knows. How does it know when to start and when to stop? That is called as gene regulation. Gene regulation is to control the exact amount of proteins needed at that time. Same picture here, eukaryote. It's completely different. Primary transcript is formed, it is processed, moves out, transported out in the cytoplasm. In the cytoplasm, the protein synthesis takes place. So again, come to prokaryotic coupled transcription and translation. That will be our main point of discussion here, and it's a very beautifully synchronized process. See here what happens. Now in prokaryotic transcription and translation, this is one electron microscope photograph, Miller's photograph. Who was Miller? I will tell you in the next slide. This photograph shows a polysomal structure and a RNA polymerase continuously being synthesized, and a polyribosomes. This is the DNA. On the DNA, you see this top length is the DNA. On the DNA at various intervals, mRNAs are synthesized. 1, 2, 3, 4, 5, 6. Maybe at this corner 7 mRNAs are synthesized from the length of the DNA, and this is a real-time actual electron microscopic photograph. And on these mRNA polyribosomal structures, they have already started synthesizing the protein. This picture is shown in a clear drawing fashion here. So DNA, this is RNA polymerase, all these are moving in this direction of transcription, and polyribosomal, that is polypeptide synthesis, has started. Amino acids are being accumulated. This is coupled transcription and translation. Have this picture in your mind for my next slides. This is a small lecture. I will not go into much detail elaborating all processes, but I want to show you the essence of coupled transcription and translation. In prokaryotic transcription and translation, the process which is coupled, at any given instance of time, they are not separated. It goes on. Sometimes what happens, the ribosomes and the RNA polymerase, they may collide. There may be a, what you can say, accident. They may just collide with each other. They may not collide with each other. What happens in these conditions? We will see. And first of all, I want to have this picture of coupled transcription and translation fixed in your mind so that you will understand what I want to say. It's a beautiful process. Transcription which is going on and translation which is going on simultaneously, as you can see, 7 to 8. This allows many number of proteins being synthesized. First point means 1, 2, 3, 4 proteins are synthesized, and each protein can be synthesized in large quantities by recycling of the ribosomes. Means you see, if at this end this reaches here, dissociates, again comes, again goes on and on and on. So it's a highly efficient process. This allows the cell to make lot of proteins very quickly, and it is facilitated by the polyribosomal structure. So before going to 2 to 3 points, only 2 to 3 slides are remaining right now, but you have to understand, focus on what I say, listen very carefully. I will go to that, but understand, let's prepare for what we are going to listen. For preparation, you should know the comparison of gene expression between prokaryotes and eukaryotes. Just as I have said, prokaryotes, one type of RNA polymerase, there is only one type, it synthesizes all types of RNA molecules. In eukaryotes, there are 3 different types of RNA polymerases, and they synthesize different 3 classes of RNA, 3 classes of RNA polymerase, type 1, type 2, type 3, they exist. Second point, mRNA is translated during transcription, and mRNA is processed before translation. Whatever the mRNA is formed in prokaryotes, that is the final transcript. But in eukaryotes, it is a pre-transcript. Introns are excised off, introns are joined, and it is the processing. Then there are three levels of processing: splicing of introns, 5-methyl guanosine cap being added, and the polyadenylate tail being added. Third point, genes are not split in prokaryotes. There are genes within genes, overlapping genes are there. They are contiguous. What is contiguous? Just like continuous, there is a genetic word called as contiguous. Actually, it may be a mathematical word. Next or near to, next and near in time means if this is one, next, near in space and time, they are very near to each other. Near with respect to space, near with respect to time, without losing any time, they are very near to each other. Okay, or in being in actual contact with each other, that is contiguous. Genes are contiguous and overlapping genes are there in the segments of prokaryotic DNA. Genes are split, they are not contiguous. There is one part of a gene, here there is another part of the gene, DNA is there in the middle. Then what is this DNA? This is a junk DNA. Means if there is one gene, that one gene part is here, the second part is here, then some DNA, third part will be here. So one gene, blank, junk DNA, second part, blank DNA, third part. So when a mRNA is formed, that mRNA removes this junk DNA in the form of introns. So they are not contiguous, they are split genes in eukaryotes. They are contiguous in prokaryotic genes. mRNAs, therefore, as they are contiguous, they are polycystronic in prokaryotes. What does polycystronic mean? One mRNA encodes for more than one type of proteins. Just see the figure here. See the figure in the previous slide here. This one DNA, follow the laser pointer, this one DNA is capable of synthesizing various mRNAs, and this one mRNA itself, one mRNA, if you see this one mRNA, of course 1, 2, 3, 4 genes, different genes are synthesized. I am talking about one gene. This one gene, one mRNA which is synthesized can produce more than one type of proteins. One mRNA capable of getting translated into more than one type of protein is called as polycystronic mRNA molecule, whereas in eukaryotes the mRNAs are monocystronic. One mRNA, one gene, one polypeptide. So that is the difference. Now come to the most interesting part, the coupled transcription and translation in prokaryotes. This historic electron micrograph of transcription translation which I just showed you, this is the same one, is an operon model given by Miller and Hamcallow in 1972, and this is a E. coli image. Miller, he is very famous for his electromicrographs. Okay, because in early 1970s, Miller, he published this for the first time, and the world came to know that there is a coupled transcription and translation process. He published this electron microscopy image which showed that ribosomes, they strongly associate with the translating nascent mRNA in a concatenane fashion. Concatenane. See this word, concatenane means together in a chain or series of polysomes. Concatenic coupled transcription and translation. So what is CTT, or what is coupled transcription and translation in prokaryotes? Transcription and translation can be coupled. This was first proposed by scientist Stent in 1960s. He proposed a theory, but for many years, for around a decade, it was not been able to prove until in 1970s Miller started publishing micrographs of such photographs of electron microscopy, and then we believe that yes, there is a coupled transcription and translation. Nascent transcripts. Why this may be there? Because nascent transcripts. What are nascent transcripts? These are mRNAs which have not been released. They are unable to release because of an active force which is active force exerted by the ribosomes can release them. From where does these ribosomes comes? DNA getting transcribed, there is a mRNA being synthesized. Now this RNA polymerase, we have seen RNA polymerase can be released by row dependent and row independent type of termination. Now there I had told you two things. One is row dependent, is row molecule itself. and second is row independent is formation of a hairpin loop structure. This hairpin loop structure and row, focus on what I am saying, listen carefully. Row dependent chain termination of transcription in bacteria and row independent, which is dependent upon row factor and the hairpin loop, both are somewhere or the other associated with the ribosome, which is translating this mRNA just being synthesized. Ribosome comes and introduces a force. Active force exerted by a translating ribosome is necessary to release the mRNA from the templates, and this hairpin loop like structure which is formed is one of the reason where this translation gets terminated even in this process. For graduates, if you are listening properly, you will understand it. As I said, just let's raise the bar to a higher level of understanding. Imagine a coupled transcription and translation. RNA polymerase is synthesizing mRNA, and from this end the ribosomes are coming. So this is moving, long chain is formed, this is also moving. If this slow down, this will catch it up. If this is fast, this is slow, this will lag behind. This is ribosome, this is RNA polymerase. Think about it, I will show it. So Stent, for the first time, he discovered that translation and transcription could be coupled in bacteria spatiotemporally, means belonging to both space and time. In space and time they are coupled, they are just behind each other. Miller, he showed it with the photographs. Recently this has been also attributed to all the archaebacteria, even all archaebacteria, prokaryotes, they show coupled transcription and translation. And this is the widely accepted dogma of prokaryotic gene expression, that transcription doesn't wait to get completed for translation to begin. Okay, so now see here, this picture right now, all these A, B, C, D, E, F pictures may find somewhat complicated, but now I would like to focus on some. Let us see these pictures one by one, and I have A, B, C, D, E, F 5 points to tell regarding this. Okay, so I have one terminology here which I call it as transcription elongation complex. Transcription elongation complex, similarly translation elongation complex, and all this will be called as expressosome, expressomes. Okay, expressomes means coupled transcription and translation coming together for expression of protein synthesis, gene expression, that is synthesis of protein. So let us just focus here. Let us just focus here on A, first part. See, only one figure at a time we are going to see. A, B, then C, then D, E and F. Okay, the ligands, if you see, this yellow molecule is RNA polymerase, the green molecule is DNA, then this purple and pink molecules, they are the 70S ribosomes, means the purple is 50S, violet is 30S. Then there is a NUS gene, protein, NUS, NUS. It is an anti-terminator. What is anti-terminator? Which will not allow termination. And there is a chain of amino acids. So one at a time, I want you to focus one at a time here. Let's go to A. What is there in A? See here. So see here, A. Transcription independent translation independent transcription. What happens in eukaryotes? Normal. This is first picture. So there will be less words spoken from my mouth, more for your eyes and mind to think over it. So A is translation independent transcription. This proceeds forward in bacteria. If it is eukaryote, this will continue and this will be released. But we are seeing the prokaryotic one, B. What happens? This is now a coupled transcription and translation. Ribosome association is there. mRNA is synthesized, and the first step, this is very few amino acids, first few amino acids. Compared to C, there are many amino acids. First few amino acids. Ribosome is associates on the Shine-Dalgarno sequence. Recall it. This ribosome association is followed at the terminal end of the mRNA by binding of the ribosomes at the terminal end. Ribosome association takes place at the terminal end of the mRNA and polypeptide synthesis starts. Move further. Now this is a physically uncoupled expressome, means what is happened, RNA polymerase is going, but ribosome is trying to catch up with the RNA polymerase. It's a coupled process. Figure C. Polypeptide chain has increased, it is going towards it. If, let us say, evolution has made a facility, if protein synthesis is now not needed, this ribosome will collide with the RNA polymerase, this yellow structure, and all will dissociate. Coupled transcription and translation dissociates. But let us say, due to some reasons, RNA polymerase speed has decreased and ribosome's speed increases. What happens in this situation is, you can see here, I will come to A, B and C you have seen. Let us come to D. What happens? This ribosome, it is trying to catch up with the RNA polymerase, and if both come in contact with each other, then there will be dissociation of all. Ribosomes will be dissociated, RNA polymerase will be dissociated, coupled transcription and translation will be lost. So instead, in prokaryotes there is a NUS gene, NUSG gene product, which is a protein, and it has a beautiful function of two arm-like structure, where on the one arm it holds the ribosome 30S subunit and other arm it holds the RNA polymerase, and it doesn't allow it to attach. So what happens now? At this point, process of transcription is going on, process of translation is also going on. At this point there will be two probabilities. If both are in homeostasis, means in synchrony, if both are having the same speed, then there will be a same speed of new mRNA passing through the 30S subunit, and the same speed amino acids will be joined to form a polypeptide chain. But if there is, let us say, a faster transcription and a slower translation, this happens, F figure happens. What is happening when there is a faster rate of transcription, means mRNA moves in a faster rate as compared to translation, then at this time there is a looping of the mRNA. This looping of the mRNA may contain stem and loop structures. If there is stem and loop configuration formed in this loop, then this will be terminated. My dear friends, remember this point. Looping of the mRNA due to internal complementary sequence in the coupled transcription and translation process is responsible for a new point here, responsible for regulation of gene expression, and we will see this in tryptophan operon whenever the discussion may arise. Okay, so these are the 5 types of probabilities. Now just see, if you just see the whole process again, once again, A, B, C, D, E and F. This is NUSG is bacterial N-utilization substance. N-utilization substance. Okay, it is called as N. What is N? N is anti-termination. If this was, see the figure F, if this ribosome collides with or physically comes in contact with RNA polymerase, there would have been termination. To avoid that termination, this protein is there. Therefore, it is called as anti-termination protein. Got it? So let us see the points associated with this most exhilarating process in bacteria. Some theoretical points which I have just explained. Now in the genetic jargon, if you want to understand more, see A. Transcription initiates over 5' proximal coding sequences, eventually forming a transcription elongation complex A. Okay, A. Now B. After considerable elongation, the leading ribosome associates with the nascent transcript and initiates translation. B. Then RNA polymerase pauses, allowing the leading ribosome to catch up with the transcription elongation complex. RNA polymerase pauses, allowing the leading ribosome to catch up with TEC, forming a physically uncoupled expressome. Now it is not attached, but there is a chance of attaching or collision. But at this point there is physically uncoupled expressome. D. As the proximity, means the distance of RNA polymerase and the leading ribosome decreases, NUSG gene associates with both machineries. NUSG, N-utilization substance, it associates with both machineries, forming a NUSG bridge. This is a NUSG bridge between ribosome and RNA polymerase, forming a coupled expressome. E, if you see the E, there can be a collision. If this doesn't attach, transcriptional roadblocks can reduce the length of connecting mRNA, giving way to collision with or without G, and this will terminate the whole process. To avoid this, this anti-terminator acts like this. Alternatively, you can see either it depends upon the rate, it can allow with loop or without loop. Translational slowdown, if translation is slow and transcription is fast, translational slowdown lengthens the length of the connecting mRNA, and in this case it loops out of the complex, and these processes are reversible. If it gains speed, then the loop will be removed. Okay, so this is how the coupled transcription and translation takes place at the molecular level. It's a very beautiful process governed in the bacteria. Bacterial N or N-utilization proteins are transcription anti-terminators, and these 3 points you should remember very importantly. Many bacterial NUSG homologs have been discovered which also possesses anti-termination activity. NUSG is not the only protein, their homologs means NUSG-like proteins. Their name itself is NUSG-like proteins or NUSG similar proteins, homologs. They have been discovered, and they all have a point in controlling the coupled transcription and translation process in anti-termination activity. NUSG, it aids in Rho silencing the transcription. Rho silencing, as I said, NUSG is also involved in making the Rho inactive, especially where there are damaged DNA and damaged RNA, due to which there may be a diseased protein, maybe protein responsible for some disease. If that is probability of being synthesized, then NUSG doesn't allow that Rho to act. Okay, so NUSG cooperates with Rho to silence the transcription of aberrant RNAs, and this is an essential function of E. coli NUSG. I have given a link here. There is a full length paper regarding this. You can read and enjoy the whole process. Finally, the last thing which I would like to tell you here about the same coupled transcription and translation approaches in bacteria. I have discussed everything. I would just like to mention a point of homeostasis here. What is homeostasis? Homeostasis is any self-regulation by an organism where we tend to maintain stability while adjusting to the conditions which are best for survival, is called as homeostasis. If I want to give an example of homeostasis in humans, us, how beautifully we regulate the 37 degrees centigrade inside our body. 24/7, whole day, whole year, throughout your lifetime, body struggles to maintain the normal body temperature, which is 37 degrees centigrade. That mechanism is homeostasis. There are many such reactions in the body. Insulin, whatever amount of sugar you eat, body tries to maintain what is needed by secreting insulin. It tries to regulate, govern. That is homeostasis. Here in this process, the transcription and translation, these are the same pictures. You see in this picture, the nucleotide and amino acid homeostasis is taking place, is being controlled in coupled transcription and translation. Nucleotide homeostasis, nucleotide and amino acid homeostasis. From the nucleotide pool, nucleotides are being added, amino acid pools, tRNA, they are bringing the amino acids, and at the same time requirements are there. It is continuously balanced. Under some faulty conditions, under some starvation conditions, this balance is disturbed. I will just mention that. This is the last point in this discussion, but the imbalance is not common. Homeostasis is common. Maintenance of balance is common. So you see here figure A, this is physically coupled transcription and translation. Under the conditions which slows down the transcription and the elongation rates below the translation rates, I have given this example. Translation is fast, transcription is slow, both come and join. At this point what happens is that this RNA polymerase was moving in this direction, but as it comes and collides, it pushes the RNA polymerase back. That is called as backtracking. The reverse sliding of RNA polymerase along the DNA and RNA is called as backtracking. Okay, the leading ribosome catches up with and physically pushes the transcription elongation complex, that is the TEC, forward, equalizing the transcription and the translation rate. So as this is moving, backtracking is reversible sliding of RNA polymerase. And in normal conditions, if backtracking is not there, means if the RNA polymerase is having the same speed as that of the ribosomes, then they will move at the same, this will push it to moving. But if this is fast and this is slow, then there will be backtracking. So backtracking has been connected as an important process in bacteria, and eukaryotic cells also backtracking is found, and it is one of the fundamental process in controlling the transcriptions, elongation, pausing, termination, high fidelity of the process, genomic instability, all these are governed by backtracking. Now B, if you see here B, the RNA polymerase is far way behind, and ribosomes are far way behind the RNA polymerase. Why this thing happens, or when this thing happens? When translation elongation lags. Translation elongation is polypeptide chain synthesis. When translation elongation lags behind transcription, it lags behind transcription. This happens when there is nitrogen limitation. Nitrogen source is very important for the growth of a cell. When nitrogen limitation takes place, then this thing happens. I am trying to tell you the bacterial approaches for during coupled transcription and translation. What happens when there is oxidative stress conditions? Oxidative stress conditions are generated like when hydrogen peroxide is there. So those bacteria which have H2O2 degrading enzyme, you have studied in your lower classes. Okay, H2O2 broken down into H2O and O2 by peroxidase enzyme, hydrogen peroxide degrader peroxidase enzyme. So this such type of oxidative stress condition, there is an increase in the PPGPP, that is guanosine triphosphate molecules, and this results into even low nitrogen condition results into a lag, and this gap is formed. C is homeostasis. You can see when slowing is there, slowing of transcription and translation are there, then there is a particular equalizing of transcription and translation also possible if at that time sufficient amount of amino acids and nucleotides are added at this growing chain. So slowing down the transcription, elongation, transcriptional elongation, promoting basal translation, and balancing the nucleotides and amino acid stocks in the nucleotide pool, and the amino acid pool is maintained here, and that is called as homeostasis, which is a fine balance of the two macromolecules. So I hope you understood the today's discussion, which was coupled transcription and translation in bacteria. This is one of the most interesting topics in protein synthesis when it comes to prokaryotic perspectives. I hope I have just taken this up to a higher level, for maybe a bit hard for the graduates, but I request you to take as much as you understood. Try to explore newer domains of knowledge. Don't restrict your thoughts only to syllabus. Syllabus is just for your guidelines, and the remaining world of knowledge is yours. How far you have to go? Sky should be the limit. Aim for the sky. You may not get the stars, but you may not get dirt and dust either. So your aims should be high. Always go for the next level of understanding. Next level of understanding will, and the next and the next and the next, will always take you to such higher domains where you need not be worried about the basic processes. That is what a student's life, mind, brain and aim should be. So here completes our first unit. Next topic, next unit will be DNA repair and mutagenesis. There is a huge chance when this DNA replication, transcription and translation are going on, chances of mutagenesis is very high. There are physical mutagenic agents, there are chemical mutagenic agents, there are metabolic errors and the metabolites which can cause rise to some analogous compounds which can give rise to mutation when metabolism is at its peak. Okay, all these things, and remarkably bacteria contains or has evolved into a mechanism of repair. It can repair by itself. So these much more interesting topics we will be exploring in our next lectures. Till that time, happy learning, happy knowledge gaining, be wise, be more powerful. Thank you very much.